Hello students, today I will discuss about the model of recombination. As you know that through sexual reproduction, the genetic makeup of the offsprings are getting changed from the parents by independent assortment and by recombination. So recombination is a process by which the genetic information is getting changed within the chromosome. There are different types like homologous genetic recombination as well as site-specific recombination. In these two cases, there are some specific sequence, homologous sequence is maintained. That's why it is called as legitimate recombination. But in case of DNA transposition, no such homologous sequence is present.
Ma'am, what do you mean by topology in recombination?
The topology in recombination involves the intramolecular as well as intermolecular aspect. In case of intermolecular aspect, the single or double crossover happens in between two double-stranded linear DNA or in between one double strand and one circular DNA molecule. In case of intramolecular recombination, actually some deletion happens because two sequences are present in one chromosome. Barbara McClintock during 1940s, she actually found the presence of transposon within maize. Robin Holliday in 1964, he actually discovered the model of homologous recombination. And Brian Sawyer, he actually proposed the model for Cray-Lock's peace recombination system. The recombination significance include the specialized DNA repair, specialized DNA replication, gene arrangement, gene diversity during the embryonic development. Hypothesis: during 1930s, Bayling have proposed the copy choice hypothesis, where the enzymes are jumped from one chromosome to other, but that particular hypothesis was not taken further. Then breakage and reunion model. First, the apparent reciprocity where no loss of genetic material was accepted. Then non-reciprocal cross also happened within the host and the bacteriophage chromosome, but general reciprocity, that is the production of hybrid DNA, according to the Holliday model, was most accepted model. Gene conversion. Certain portion of the gene is copied from the mother strand to the daughter one without deletion from one particular chromosome. And negative interference. Suppose one particular recombination happens in one area that inhibits further recombination at that particular area. Genetic mapping is a type of linkage mapping by which the distance between two genetic markers can be calculated with the help of recombination frequency. That is a frequency theta that helps to designate the distance of two genetic loci. 1% recombination frequency is equivalent with 1 centimorgan distance. Now, within the genome, there are some area where the rate of recombination is elevated. So, that particular area is called as recombination hotspot. In case of homologous recombination, extended sequence homology is present in between the chromosomes or within one particular chromosome. It may be happen in bacteria for DNA repair where replication fork stalls. In case of eukaryote, for the sake of cell division, replication as well as repair. In case of some virus like herpes simplex virus, it is needed for the evolution. In eukaryotes, the process actually happened within the germline cell for meiosis. During prophase 1, the tetrad is formed, the chiasmata is formed. Fine. Now the crossover happens, and during this crossover, that means the recombination happens. Ultimately, the reassorted, the recombined chromosomes are sent towards the daughter cells. Now, model for the recombination. In case of eukaryotic cell, mainly DSBR pathway, double-stranded break repair pathway, according to the Holliday model, is well accepted. Other than these, SDSA pathway is also there, that is synthesis dependent strand annealing pathway. In these two pathways, some earlier steps are common, but after some time they were diverged. In case of DSBR pathway, crossover product is produced, but in case of SDSA pathway, non-crossover product is produced. SSA pathway, that is the single strand annealing, is another pathway. Steps involved in holiday model. First, single stranded nick is produced. So that will be wrapped by rake A. Then the strand inversion or strand displacement, branch site or chi structure production, branch migration, then resolution. From this picture, that is well documented how the process is going on. In prokaryotes and bacteria, that actually runs through rake BCD pathway. So rake BCD pathway means rake A, rake B, rake C, rake D gene product, they are actually getting involved. During the course of replication, when DNA pol 3 encounters a gap, then and then it will stop over there. Then the double-stranded gap is created. That particular gap is recognized by rake BCD enzyme. Rake BCD is a kind of exonuclease as well as helicase enzyme. It will unwind the DNA and chew it up. But you know that the 3' template is chewed more than that of the 5' template. In case of 3' template, when it encounters a chi sequence, then the chewing up will be stopped. Then the rate in case of 5' template will be faster. Fine. And then again it will get chi sequence over there and it will produce a 3' hydroxyl overhang which will be wrapped cooperatively by rake A protein. Rake A protein is very important protein, you know.
Ma'am, what do you mean by chi sequence?
Very good question. Chi sequence, that is GC, TGG, TGG sequence, that is frequently distributed, the 500 to 1000 times. And you know that rake A has several different functions. With the help of ATP hydrolysis, it is actually involved in the branch migration, strand inversion, as well as it is a protein needed for SOS repair system. Then what happens? In next case, rake A is a kind of driver protein. It drives the single-stranded DNA towards the homologous duplex, towards the double-stranded DNA. So that is called the strand exchange reaction. After that, before going into the branch migration, I will tell you that there are so many other proteins in eukaryotes like chicken, mice and human. There are some rake A-like protein as RAD51, RAD52. They are also participating in recombination as well as recombination repair process. Let's talk about the branch migration. So what is happening here? The rake A bound single-stranded DNA is proceeding towards the double-stranded homologous DNA, fine, and it is actually displacing one strand, so that is called strand inversion, fine. It will ligate over there, creating a hybrid junction that is called a chi site, holiday junction, fine. So what is branch migration? So here you can find one strand having complementary to two strands. So with earlier complementary bonds will be broken and new bonding will be made, will be formed, until and unless it is the homologous sequence will be completed. Then the branch migration will be happen, a spooling action, fine, from 5' to 3' direction. That is actually happen with the help of ATP hydrolysis, with the help of rake A as well as another protein, RUVB protein. So after that, when such branch migration will be completed, then resolution will be happen, okay, with resolvosome complex, RUV-ABC. RUV-A will recognize the hybrid junction. It will recruit the RUV-B. RUV-B has branch migration action as well as it will help in the resolution process. It will then recruit RUV-C. RUV-C has endonuclease function. It will cleave the hybrid DNA and ultimately it will recombine and ligate with the help of ligase. So ultimately the recombined DNA will be produced. You know that there are some circular DNA molecule which will recombine by this process and it will make a 8-like structure. Ultimately they can finish with 2 same side circle or 1 larger composite circle. And by endonuclease action from 8 structure you can have Greek letter chi structure. Let's start with site specific recombination. So this site specific recombination needs 20 to 200 base pair specific sequence as well as some other accessory proteins. You can find for the sake of genetic rearrangement as well as during the course of viral and plasmid DNA replication. And site-specific recombination needs help of some specific enzyme. Here I will show you the model for integrase that is actually happen in case of bacteriophage lambda. Now integrase is a type of recombinase having tetrameric structure. First it will find the specific sequence and it will make a cut over there. Fine. There is a specific tyrosine residue within the recombinase or integrase. So a phosphotyrosine bond is made. Without the help of ATP, this particular high energy bond will supply the energy for the second cut in the same DNA. Fine. Then ultimately these two steps will be again repeated and with the help of recombination it will produce the recombined product after ligating with ligase. Next, the effects of site-specific recombination can be found where one strand is linear, other is circular. So, you can find the insertion as in case of bacteriophage lambda, or the sequence will be exchanged when two linear strands are present. You may find within one particular strand, if two site-specific recombination sites are in same direction, they will give rise to the deletion of the intervening sequence. But if they are facing to each other, the intervening sequence will be inverted. Next one, occurrence. You can find in case of integration of lambda phage to the E. coli chromosome. Lambda phage must have ATP sequence having 15 base pair homology sequence with the ATB of bacteria. After integration with the help of int protein at R and at L, these two sequences will be produced. Fine. At the overhang region. Ultimately, in the same process, the excision will also happen by the help of exis protein. And in both the cases, some integrating host factors, IHF, are helping them off. You know that 515 base pair sequence is homologous in between at P and at B. Next, you can find the presence of FRT-FLP system, the recombinase system in case of Saccharomyces cerevisiae. They actually also pass through the site-specific recombination, and you can find in case of bacteriophage P1 the presence of Cre-Logs-P system. You can find some specific inverted repeats over there. Okay, and you can find within the FRT-FLP system ABC specific consensus sequence, 13 BP sequences are present over there. Transposon. So apart from which, in case of transposon, that is referred to as the illegitimate recombination. So from the donor site, some sequences jump to the target site. Okay. So at the donor site, some sequences are copied to the target site. According to the type, they may be the simple one. In case of IS element, bacteria IS element, this sort of IS element, they possess the transposase enzyme gene. Transposase is very much needed for the transposition. Next, the complex transposon. It may be the composite having two IS element, but in case of non-composite transposon, no IS element present. But antibiotic resistance genes, resolvase genes, all those genes can be present. In case of retrotransposon, that is mediated via reverse transcriptase action, RNA. Okay, the process is quite different. Now let's talk about the general pathway of the transposon. First, transposase enzyme makes a nick at the donor site. So 3' hydroxyl end is getting produced. Then the transposase make two staggered cuts by making two sticky ends at the target site. Then these 3' hydroxyl end can make the nucleophilic attack, and ultimately they will be ligated over there, they will join over there by duplication at the target site. But in case of replicative transposon like complex transposition, the co-integrate is made. Fine. So that particular co-integrate can be resolved by resolvase enzyme. And you know that in case of retrotransposition, that will be mediated via reverse transcriptase as well as by non-homologous enjoining process.
Ma'am, what are the applications of natural recombination?
Very good question. There are so many applications of the natural recombination in eukaryotic system as well as prokaryotic system naturally. In case of bacteria, for transformation, for conjugation, transduction, it is very much needed. In case of eukaryotes as well, it is needed for the gene shuffling, DNA repair, as well as it is needed for the gene assembly to produce the immunoglobulin structures as well.
So, ma'am, are there any in vitro applications?
Of course, there are so many in vitro applications of recombinations are there. In case of protein engineering, you can find the parental protein subunit or fragments can be swapped. They can be changed with the help of such recombination. And in case of cancer therapy, the recombination is also used because there are so many proteins called BRCA1, BRCA2, they can interact to the RAD51 needed for the recombination purpose. The mutation in all those genes, they actually cause some sort of cancers. Now, against all those cancers, the drugs can be generated. So, all those things are needed. The recombination is very much needed in case of cancer therapy. So, PRP1 inhibitor, they actually interact with the BRCA1 and 2 genes. The very important role in case of repair is generated via recombination. You know, recombination repair is very much needed in case of prokaryote as well as eukaryotic systems as well. Let's talk about the single-stranded break repair. You know that when DNA pol 3 proceeds via the replication, okay, it may encounter a single strand lesion or a single strand nick. If it encounters a nick, then REC-BCD pathway will be operated. Okay, REC-BCD exonuclease helicase activity, it will recruit the REC-A. Fine. But if REC-BCD pathway is mutated, then REC-A4 pathway will be operated. When DNA pol 3 will encounter a lesion, then REC-A4 pathway will proceed via producing a chicken foot model. Okay, a retrograde pathway, a chicken foot model. Then what you can find in case of REC-A4 pathway, REC-Q has helicase activity, REC-J has nuclease activity, it will chew up, fine, and it will produce a 3' overhang, which will be with the help of single stranding binded protein, it will recruit REC-A once again. Ultimately, they will recruit REC-A with the help of REC-A4 system. And once the REC-A will be engaged at the 3' hydroxyl of the single strand nick, ultimately it will go for the branch migration, strand inversion, branch migration, then resolution. Then it will follow the common pathway. So, REC-A4 is another pathway by which single stranded lesion can be repaired. Double-stranded break in the homologous DNA. That is quite infrequent, but it will also be repaired by the help of double-stranded break repair holiday model pathway. Fine. Again, 2 overhangs will be made by single strand nick because nicks are already made. Fine. So, they will be recognized by the REC-BCD, then it will recruit REC-A, then 2 junctions, 2 holiday junctions chi structure will be made, and ultimately they will be resolved to produce the transformed product. So, you will get the recombined product at the end of the repair. Next, importance of the recombination repair. As you know that in case of eukaryotic system, the REC-A-like protein is RAD51. RAD51 is interacting with BRCA1 and BRCA2 gene product. If any mutation happens in these 2 genes, ultimately it will lead the system towards the prostate cancer, ovarian cancer as well as colon cancer. Deleterious effect of recombination repair. You can find in many cases, the dimeric cointegrate DNA can be produced if any mutation happens. So, this particular dimeric DNA can be resolved with the help of XCR CD system present in E. coli, with the help of resolvase as well, because can you recall all those things, because in case of cointegrate, in case of transposon is resolved by the resolvase, as well as resolvase is also applicable to the site specific recombination, because integrase is a package of both endonuclease and ligase. Okay, next one, the mechanism for the gene assembly or gene rearrangement in case of kappa gene of undifferentiated B cells. So, to produce the proper IgG light chain kappa, some gene assemblies must be there with the help of these recombination. So, what is going on? You know that there are 300 variable region, 4 J region, joining region, and 1 constant region for one particular immunoglobulin. For proper signal, when proper IgG molecule is to be produced, then what will be going on? There is a specific sequence called RSS, recombination signal sequence, okay, just after V and before J, specific sequences. What is going on? There are other 2 proteins, RAG1 and RAG2, recombination activating gene. So, all those proteins, they will recognize the RSS sequence. Fine. First, after V region and RSS sequence, RAG1 will make a cut. Fine. So, 3' hydroxyl overhang will produce. It will, via nucleophilic attack, it will attack to another RSS just present before the J region, specific J region. So, a loop will be getting produced that will be deleted, then with the specific V, specific J will be joined by the ligase. So, in this way, in the undifferentiated B cell with proper variable region, the proper joining region will be attached through this sort of recombination process. And very important in vitro process is running through your recombination, that is the genetic manipulation in animals. So, transgenic animals are produced. In case of transgenic animal, all cells having new combination of the genetic makeup. So, it can be happen either via homologous recombination. So, what is going on in case of homologous recombination? The target gene is there. So, through gene targeting and gene disruption, that will give rise to the transgenic animal. Gene targeting means displacement of the exon either or endogenous sequence, and by gene knockout, it will be disrupted. So, there is a specific target gene, fine. It will carry a positive selection like neomycin resistance. Okay, so we will apply a drug for positive selection, that is G418, that is the member of neomycin, and for negative selection we will put thiamidine kinase, TK gene. Okay, if homologous recombination does not happen, so it will carry the TK gene, so against which we will give the drug ganciclovir. So, in this way, positive and negative selection, we will select the recombined gene where the gene will be knocked out or knocked in, and ultimately they will be injected in embryonic stem cells. Other than these, Cray-Loxp system can also be taken to generate the knockout mice, because the Cray is very specific towards the LogSP sequence. So, within LogSP sequence, if your target DNA is inserted, so that will be deleted. In this way, the knockout mice can be generated. As you know that there is an evolutionary origin of all those recombinational protein which are engaged in recombination repair as well. In case of bacteria, E. coli, you can find RAC-A. In archaea, you can find there are so many other proteins, RAD-A, RAD-B, etc. As well as in case of eukaryotes, in case of human, chicken, mice, you can find RAD1, RAD2, DCM1, etc. So, they are all in the emerging pathway. They are the footsteps of the evolutionary origin of recombination. So, students, I have discussed about the model sequence, consequence, and several different significance of the recombination with respect to different systems. Thank you.
Thank you, ma'am.
Thank you, ma'am.